Pero no habían parlato.
Okay, it's a real pleasure to have with us Roberto Silva Andrade. It's impossible to summarize in less than 1 hour the experience of the speaker, so I will refrain from doing that. Just to tell you that he comes from the Instituto de Fisica of the University of Salvador, in Universidade Federal de Bahia in Salvador, Brazil. He's, among other things, responsible for Flora being here, and also other people. Professor Andrade got the PhD in Germany, in Regensburg University, doing some work on turbulence modeling by chaotic nonlinear dynamic methods. And also he spent some time in Brussels, in Université de Bruxelles, with a group of Eugene and others, doing some chemical oscillations. And he has stayed in many places in Europe and in America, but then he returned back to Brazil, to Bahia, where he's leading an interesting group who is working in the group is working in many things which are strongly related to work here. This conference will be about some complex networks, and he has done a lot of works in analyzing the, say, modularity and unities and stuff in complex networks, as applications in biology. He has also done some network analysis for climate of meteorological time series, general statistical mechanics, spin systems, disordered systems, and everything you can think on statistical and molecular physics and complex systems is in some way related to the work of Roberto. So we're happy to have you here, and the stage is yours.
Thank you, Emilio, for this opportunity of being here. Two reasons: for taking part in the piece of Florida, and also for having the opportunity to deliver this talk. The title has been already announced. It's too long to be repeated again, so I will not repeat it. These are the logos of the institutions that have participated in this work. This is the logo of my university. This is the logo of our group. The name means stays in Portuguese for Física, Estatística e Sistemas Complejos. So this is our group. This is the group. This is the logo of the University of Feira de Santana, and this is the group of the microbiology in Feira de Santana that have been together in this work. This work has been. This is the team. At that time, Charles, who is here, was there. Okay, and so he is the team. These are the team from the Department of Biological Sciences in the Feira de Santana University. This guy is from, he's a Brazilian guy, that's his name, but he's in the Biology Institute of the Federal University of Bahia. And Thierry is also in the Mathematics Institute of our university. Thierry belongs also to our group, to the IFISC. Okay, so let me briefly say about the outline of the talk. I will present two slides on our group. Then I will insert some slides on basic concepts of network, and then we start with the subject that has been the object of this research, something about phylogenetic classification and proteins, protein networks, our results and the conclusions and perspectives. Okay. So this is our group, the task. There are 5 faculty members in our institute, myself here, and then Ernesto, who is the guy of the Polytechnical School, José Garcia Vivas Miranda, he is from our institute, he has been here to this place. Suani, she is also in our institute, and Thierry, as I said before, he is a physicist who went to mathematics and stays in between mathematics and physicist. These are the research lines that we do in our group. We have done some work on statistical equilibrium, statistical mechanics. Well, particularly, we are interested in magnetic system, spin systems, in some kind of networks where we can control disorder and periodicity. There are some work done also in the foundation of statistical mechanics, here specifically on the non-extensive statistical mechanics. Ernesto Borges is a guy who is doing work on this theory, on this subject. Then graph theory and complex networks. Graph theory is more from a mathematical point of view, and complex networks from physics point of view and interdisciplinary point of view. We also do some periodic sequences together in a periodic magnetic system, cellular automata. Some working on scaling variance in time series, and we have been using this to analyze rainfall data. We have been using to analyze seismograms and oil also, and also quite recently we have also analyzed some economic time series, and of course we have doing here also some biologically motivated models. This then is what we do. Of course it is, we do not do this every time, all the time, but okay. I included here some concepts of network theory for the sake of completeness. The formal description of, we would say, formal mathematical definition of a network is a set of elements called vertices or nodes, and links that connect two nodes of a vertex, two nodes of the graph. Okay, so the graph is set as can be represented very compelling to have a visual representation that is some series of dots and connection between those dots. So you can go and use your vocabulary to say unlabeled, edge labeled, vertex labeled, vertex colored, edge colored, vertex and edge colored. These are just possible ways. When you say color, that means you make an attribute to specific attribute to the node. Sometimes it's important. When you put some color to the edge, that means you put some attribute to the edge. This attribute to the edge is important in the sense that we are going to talk, because we associate a weight. You can weight the edge. You can also, the weight that we will use here will be the kind of similarity between two proteins. Okay? And then so you will be most concerned with graphs like this, where the nodes are labeled and where you can put some properties on the edges. Of course, there are also some things like that. Here you have the opportunity of putting two connections between the same pair of nodes. We are not going to consider this. We not consider self-interactions. This something that we can consider is disconnected graphs where one node is not connected to the rest of the main graph. And also you can have some directed graph where the connection between two links has a specific direction. We will not consider this. We will consider an undirected network. So you can build networks in several graphs in different ways. Here just representation of those. This is very scary. It's not the throat of that animal that swallows. It's just an Apollonian network. Okay, but you can do nice pictures with networks. You can do very interesting things. You can then talk more formally, you can have some deterministic networks, like the KD tree, like the Apollonian networks. So the Apollonian networks have been I have been working with it in several models and scheme models, and also as network as such. And it's very convenient because it's a way that you can draw it in a very precise way, and you can use this kind of network to compare the results of what you get in other randomic networks. Okay. Then you have can talk about the random networks where the links are set to a kind of degree of randomness. Okay. This was first studied systematically studied in the 60s by those guys. That is this guy. And then in the last decade, a number of implementations have been appeared. This is the protein network, the representation of the protein networks. One of the first ones. Has been many others representations more complete than this one, emails and so on. This is just for the purpose of calling the attention to that many subjects have been doing with that. This would be the internet, the IP addresses. As I said, you can do some art in this kind of this stuff. Here is and I thought there was some. No, okay. Okay, this is out of order. Then I go back to the first one. After the basic work of Erdos and Henai, there are two very paradigmatic networks that have been used. They introduced it in the late 90s and have been used ever since. One is the concept of a small world network that was proposed by Watts and Strogatz. The issue is that you are between the regular and the random network, and so you have some high clustering coefficient, and you have short paths between the nodes. This guy here, which is the random network introduced by Erdos and Henai, has a short path between the nodes, but has very low clustering coefficient. So small world stays in between. The second type of network that was introduced was the scale-free network, and this is related to the distribution of the degrees in a network. You can have a network degree distribution following a Poisson distribution. You can think about the number of neighbors of connected by roads, or a number of neighbors connected, of airports connected to other airports. Here you do not have a statistic like this. Here you have statistic like this. And this is the concept of scale-free network. Barabas and Albert, they suggested a model, as well as Watts and Strogatz. They proposed two different algorithms for generating these kinds of networks. Okay. So I will go then back, and I will talk then the basic concepts that we use to measure and to differentiate one network from the other. These are the very fundamental concepts. You use the degree, which is the number of links that are connected to the node. Ki is the number of links connecting to node I. The clustering coefficient measures how much of a node's neighbors are connected among themselves. This value goes from 1 to 0. In a tree, like the KD tree, Ci is 0 for every node because you have no closed loops. While in the Apollonian network, you have a very high clustering coefficient. You have the average minimal path to a node Li you have to consider a node I and see how many steps you have to go to reach any other site, any other node in the network, and this gives the shortest path, the minimum path between node I and node J, for instance, and then you take the average over J. Okay? Then you have the average minimum path of node I, and also you have the diameter of the network, the largest path in the network, the largest shortest path in the network. Okay? Okay, we use these concepts throughout our work. Everyone working in network uses this kind of concepts. And of course, there are much greater number of measures that you can define on a network. And some of them are defined and used for some specific purpose. We are going to specialize in our talk what are the measure, the specific measure that we are going to use in this approach. Okay? Okay, then we are, this something what I also would like to call the attention in a very pictorial way. We are talking about complex network, and so one can ask how complex is a network. And this is one way of representing the network. Okay, you have the numbers. If you have the numbers from 0 to 19, we have 20 nodes, and I just connected the nodes that are just draw the links that connect the nodes. And then if I ask you how complex is this network, some of you may think that this would be a complex network because the links are in certain way some random from one side to the other. But if you go to the next picture, you see that this is not completely, this is a completely ordered network. If you go back and you relabel those numbers, you can go back to this kind of network. Okay. Here I did not use it should be here numbered according to the labels would be renumbered. It's not according to this order. And sometimes it's very important to address this issue. If you relabel a network, you can find communities inside the network. And this is really a very tough question. How to find communities inside a network. Sometimes it's just a matter of putting the nodes together that are more close to one to other. If you use some very generic programs like the Phylogenetic program, it has a way of representing networks. It minimizes some function that puts together the nodes that are close connected. Okay, but sometimes it's not sufficient to find out the communities. But this is some issue that we are going to deal with in this talk. Another important concept is how you describe the network. We use basically the concept of adjacency matrix. Okay, you have nodes 1 and 2 are connected. So in the matrix, this is a 5 by 5 matrix. The position 1, 2 is corresponds to 1. The position 1, 3 corresponds to 0 because 1 is not connected to 3. Okay, this is the adjacency matrix representation of this network. This is another example of that network that I talked to you before. If you see the networks here, if you see through each line, you see that there is only 2 ones in each line. Okay, and so that's why it was possible to draw it in a circle. Okay. Okay. This is the picture that I told Emilio. That is the same one that you are. The representation of the tree of life. We are going to talk then about the work that we're going to then to talk about now is work that we have done to use this concept of complex network to propose a way to to find a phylogeny given some input, very basic input data. Okay, the phylogenetic trees, they are kind of periodic tables of biological diversity. You put together what fits together, like in the periodic table, you put the metals one below the others, and so you have different distinctions. You have a biological classification. This is proposed for physicists, not proposed for biological people. Okay, we know nothing much more about this way of classifying the species according to species, genus, family, order, class, phylum, kingdom, and also the recently introduced domains, archaea, bacteria, and eukarya. They have been introduced and they are come upon this more usual classification scheme. And how one proceeds with a phylogenetic classification, we go on through methods to classify it, grouping, analysis, and they first, when the people started, biologists started classifying, they were much more based on the morphological aspects of the animals. Nowadays, they input very, not only on the qualitative biological feature as well, but on very mathematics. They use some kind of substitution matrix, and they demand also a very large kind of computation to proceed with this, looking for all possible ways of classifying according to the information that we put use as input in your model. Okay. What we are going to use here? We are going to use data from protein and other molecular and other basic structures in organisms. Let me try to explain you how we chose the data. When you have an access to an open bank of biological data, there's a huge amount of data. With the sequencing and the procedure being almost full automatized, it's possible to generate a huge amount of data. And so we chose then to devise a model for phylogenetic classification using very basic information. The basic information that we are going to use is the structure of proteins. Proteins are composed by a sequence of amino acids, and then we want to compare proteins from different organisms and use the structure of protein from different organisms to provide a classification. Okay? Then the issue was to choose which proteins are going to use, and then which proteins are amenable to a comparison, a simple comparison. Okay. Then we know that there are several organic biomolecules that they are present in almost all organisms. For instance, ATP. ATP is present in every living organism. Glucose is present in a very huge number of organisms. In our work, we concentrated on one specific of those molecules, which is not glucose, but it is the chitin. Chitin is a biomolecule that is present in a large variety of organisms. The synthesis of such molecules requires the presence of several enzymes. Each of for instance, chitin requires the presence of several enzymes, and each enzyme catalyzes a step during the production of chitin. Distinct organisms use the enzyme sets, what we call the pathways, to obtain the same molecule, the same chitin. And we find out that our proposal is that organisms can be classified according to the similarity of the pathways. That means here the molecules that are used to synthesize some basic biomolecules. In the case, we are going to use the chitin. The similarity here that we use is based on protein data from completely sequenced genomes. Okay, we try not to improve the quality of the base input data, and we base our classification on what we call the distance between networks. It's not that we base our classification on this; it's that we use this concept of distance between networks as an important step in the classification scheme. Okay, the chitin, we use data from the chitin synthesis. It is a carbohydrate. It is present in cell walls of fungi and exoskeletons from arthropods. It's the second most abundant polysaccharide in nature after cellulose. Okay, this is what we are going to present here. The method that we devise can, of course, be used. If it works here, it could be used to using any other molecular synthesis, using data related to any other molecular synthesis. This is the chitin metabolic pathway. This is how it's produced. Okay, you see here that in this chart you have some the presence of some enzymes. UDP is an enzyme. This name here, acetylglucosamine, is enzyme. You have gluco is gluco. It is exo somewhere here. It is exo some. Okay, there are some other places where some specific enzymes take part. Here are the basic protein enzymes that we use as base in our comparison. We take the UDP, which is the acetyl, which is the short name for anything like this. Exo, which is the short name for this. And phospho is a short name for this. Gluco is a short name for this. Okay? Everything here ends in ASE. ASE is the what's identified as being an enzyme. All acid, I don't know how it's in Portuguese to say acid. It indicates that it is enzyme. Okay, so let me tell you now that I told you what we're going to use. We started working and we downloaded the information from Gene Bank. It was some years ago, but then we kept working with this because it's impossible to think that you will update your bank every day because you never finish, you never start the work. So you have to, not the issue that you never finish, you never start. So you have to download the information and then work with that. From this sequence that we downloaded, we found out 1695 protein sequences that were related with 13 different enzymes in the chitin networks. From these 13 networks, enzymes, only 5 we found to be representative for building a network. Why? Because the number of organisms that contained these 5 kinds of these 5 enzymes, they're abundant. I mean, abundant more than 100. Okay. The other 8 enzymes, the number of organisms in which they are present, was less than 100, and we thought it's not sufficient for building a network. So that was why we chose these 5 enzymes among the group of 13 that were available to us. Okay, Then, as I told you before, you have the enzyme, and the enzyme is composed by a sequence of amino acids. If all enzymes have the same number of amino acids, and you make a comparison between the two, you just have to compare amino acids in the position 1 of the two. If they fit, you put a 1. If they do not fit, you put a 0. Okay? And then you proceed, and you have a fixed number of amino acids. It's easy to make a comparison. But if you, what happens is that the enzymes that place the same hole in different organisms, they do not have the same number of amino acids. So you have to proceed, the comparison is more tricky. You have to see if some have been excluded, some have been repeated, some parts have been inserted, and so on. And so the comparison is more difficult to be made. Fortunately, you find in this data bank programs that proceed with this kind of classification. So we use this program, the BLAST program, that is available in the GeneVan. And this program proceeds with the classification. Okay? It's interesting to say that when you compare then two proteins, you build what we call a similarity matrix. This similarity matrix is a number, it's a weight that I called your attention in the beginning of the presentation. 100 means 100% similarity. Two proteins are completely equal. 0 bit would mean that they are not equal at all. And so in between you have the degree of similarity between the two proteins. It's interesting to call the attention that when you run BLAST, you compare protein I with protein J. But if you do protein J with protein I, the results can be different because the way they use to proceed with the comparison is not symmetric. And so, for our purposes, we decided to work with undirected network. And so, to work with an undirected network, we symmetrized the similarity matrix. Okay? And then the symmetrized version leads to an undirected network, and so to undirected adjacency matrix. Here, it's important to say, adjacency matrix, I included the definition when you have a network. So adjacency matrix is has only 1 or 0 values. No, it's not possible to have 0.5. It's a network adjacency matrix, according to the definition, can only have zeros or 1. So if you have an unweighted network, then we can construct an adjacency matrix. How you can construct?
What happens if you have a protein that is part of another? Like, I don't know, you have a piece of a protein and then you do this kind of similarity analysis. You get a 1 or you get another value in the similarity?
If one is a part of the other, yes. You probably, if you compare the short one with the high one, with the large one, you have a small value. But if you make the other way around, you will get a larger value. Okay. Then you want to use this similarity matrix to define a network. You define a network in which the nodes will be denoted by I. They represent a protein. And an edge, a link between the nodes will be connected if the similarity index is above a given threshold. Okay? This is a way of making you project the entire similarity matrix into an adjacent matrix. And if you do that, you have a network. Okay? This is the idea. Of course, if the choice you make for the threshold defines the network. Okay? If you choose the value of STH to be 0, Then you include everyone is coupled to everyone. It's very bad way to choose because you have no information about the proteins, how the proteins are made. If you choose to be 100, okay, full agreement, it's also very unwise way to do because only a few pairs of proteins will be linked together. So you have to find a best way to find this threshold value, in which, and now you call the best way is the value of STH, where you can retrieve biological information, valuable biological information. This is how we are going to proceed. To do this, we this is actually the most important step of this work. We have talked about this degree distribution, clustering coefficient, average, path length. We have this concept that I did not mention it before, the edge between us, and we have this concept that we have not talked before, the distance between two networks. Okay, this is basically we are going to use. So, as I told you before, we are going to make a judicious choice of the value of STH that optimizes the reliability of classification scheme, which is mean that it makes it possible to retrieve the biological information on the system. So let me talk to you about the distance between networks. If you have a network, you have it can be given by a adjacency matrix like this one. You can consider also what we call the L neighborhood of a matrix of a network. The L neighborhood is, for instance, represented by this matrix. This matrix, which looks like this one, but it just says that this element, for instance, 1 to 6, is not connected to direct. This one says that node 1 is connected to node 2, to node 3, to node 4, to node 5, but it's not connected to node 6. This one tells you that node 1 is connected to node 6 by a two-step walk. So we call this the second order neighborhood of node 1. Okay? So this matrix here, which looks like this one, is composed of zeros and ones. Okay? It is the matrix that says that which nodes are connected by the shortest path between two nodes is a two-step walk. And so you can also find a third, a fourth, and so on. You stop when you find the diameter of the network. Okay? Because the diameter is the largest distance, shortest distance that you can have in a network. In this case, this is the Apollonian network. It represents the Apollonian networks. The diameter is L equals 3. Okay? Then, of course, you can then multiply the L equal 2 network by 2. You can multiply by any number. You just multiply it by 2. Multiply the L equals 3 network by 3. And then we sum them. Okay? Then you have a neighborhood matrix. Okay? A neighborhood matrix is a full matrix. It's not like the adjacency matrix composed by zeros and ones. It's a full matrix in which only the elements of the diagonal are 0, and the numbers indicate how many steps one is the shortest path between two nodes. Okay? Since all information on a network is contained in the adjacency matrix, the neighborhood network just enfolds the information that is present in the adjacency matrix. Okay, it brings no more implicit information because the adjacency matrix contains all information on the network, but it enfolds the information that was here. Okay, and also it provides a good way of representing networks. Okay, you can draw chart with color codes indicating which points are connected to it, and how many steps you need to connect one node to other. This is what is equivalent if you make a representation like by links and nodes, dots and links. This would be a first order representation, second, third, fourth. And then when you put all together, you have a complete graph where all nodes are connected to each other, but the colors indicate to each neighborhood two nodes belong. For instance, node 3 and 7 belong to the second order neighborhood. Okay. If we do this, you can define a distance between two networks in our procedure. For instance, you can have you choose a given value of SGH and a very close value of SGH, and then you define two networks. Let's say one network with 30%, a network with 31%. Okay, trying to have two matrices, full neighborhood matrix of the 30% matrix of the 30% network and 31% network, and then you make a distance, a Euclidean distance between these two matrices. You just subtract, square, sum, divided by the number of elements. Okay, that's what we call the distance between networks.
Sorry, can you repeat the definition of the distance? Please repeat the definition of the distance.
Of the distance. The distance between two any networks, what we call the distance between any two networks, if you have two networks with the same number of nodes, okay, you build the neighborhood matrix of each one of them, then you subtract element by element.
All right.
Okay, square, sum, and divide by the number of elements. Then you have an Euclidean distance between the two matrices. Okay? In the case here, the two networks, the nodes are the same for the two networks. What varies from one network to the other is the possible presence of more links than in the other one. Okay? But the nodes are the same. But you can think of this by two completely different networks. You have, for instance, R12 and I network and a Bahia network.
So and the distance is a matrix as well.
It's a matrix.
All right.
The distance between the matrix element of the neighborhood matrix.
So you don't have an absolute value. Then, I mean.
No, if the two networks are completely equal, the numbers are completely equal, so the distance is 0. Okay, but you can find different values for this. In the case here, okay, the nodes have the same meaning, and the distance differs if you remove a link in the network. Okay. So if you compare the distance and the threshold value, the SGH, you find something like this. From 30 to 31, the distance is very low. And then you find some place like this, like this, like this, and then it goes here down. Okay, that's how it looks, the distance as function of the threshold. Okay?
I wanted to check directly. If you calculate the weight between, so Sij, and then you put a threshold to get a 0 or 1, why don't you take directly the Sij in one case and the Sij in the other case as a measure of the distance?
That could also be. But the structure would be different. It's not necessarily means because when you compare the value of S30 to S31, for instance, you do not get direct information of the neighborhood structure of the network. Whereas if you take the adjacency matrix and build the neighborhood structure then you get this enhanced.
Yeah, but on the one hand you are putting a threshold, so you are translating, introducing there some spurious, because the threshold is something that you don't have a criterion to. And secondly, the neighborhood is, as you said, is built on the adjacency matrix, so there is no extra information. So you are basically repeating information, so you are iterating the adjacency matrix to get the second and the third.
Yes, but
so
the adjacency
so you introduce some perturbation or some mistake in the first step, it's going to be amplified later on.
Now the issue is that the adjacency matrix has only zeros and ones. And it's very difficult to make a comparison if you only compare zeros and ones. Whereas if you use a matrix, the neighborhood matrix, you get information of the whole neighborhood structure. It's much more information that you are able to use.
Yeah, but basically you are using the same information, so in order to get the neighborhood.
Yes, but you weight in a different way, so you are able to extract more information for this. So let me tell you how we succeeded doing this, okay? And so that's the way how we interpret. Here, let me show you. Okay, here is what you get. Here you are 40%. 40% something here. Okay, you have seen no sharp peaks here. Okay, and so if you build a dendrogram of this network, we use the Newman-German algorithm for extracting the more relevant nodes with the highest betweenness degree to separate communities. The Newman-German algorithm is an algorithm for separating communities in a network. It's based on the concept of the largest betweenness. You evaluated the betweenness degree is the fraction of the shortest path between all nodes that traverse a given link. So you evaluate the betweenness degree of all links in the network, and then you extract that one. Okay? Then you do the same thing. You recalculate the structure of the shortest path and proceed like this. So at each time you have to recalculate the betweenness degree of each link in the network, extract the largest one and recalculate. It is a very suitable algorithm for finding networks, finding structure. The issue is that it's very takes very long time for doing this. So if you use in the 40%, you find something like this, you get no information. If you use at 51%, 51% corresponds to this, okay, you find this structure. And if you draw the neighborhood matrix of this, you find this kind of structure for your network, where you can easily recognize it, structures 1, 2, 3, 4, 5, 6, and so on. This blue stripe means that this node is no longer connected to this one. These three isolated groups and some more isolated here, the small ones, are probably related to these peaks here. Okay? Whereas this one makes the separation of this big block with that one. Here we can still find some 3 blocks inside here. If we proceed further, you find another one here that will probably represent the separation of these 2 ones and so on. So the structure of peaks that we find in the Euclidean distance function of threshold tell us where you should proceed with your community analysis. Okay, so we did this. The biologs of the group then identified which bacteria are these. If you use Pajek to represent, you find something like this. Okay, so you see that the structures are really presented. You see that this one is isolated.
This is related to the collision threshold in some way. Is it related to percolation threshold in some way?
It is in some way, but the issue is that you don't have only one percolation. When you have percolation threshold, you usually have one giant plus in just one transition. Here it will not find everything happening in just one point. You have some group of transitions. Okay. Here in this case you have some peaks. As I show in the next, for some proteins, for some enzymes, you find that everything happens together. So we find this, then identification, and this identification was based then on other classification scheme that we found in from the biologists. Okay. Okay, again, which are the bacteria, and so on. These represented the data we used for UTP. We proceeded with acetyl, glucohexamine, phosphor, and you see the picture is not the same. We have STH at different values. Here is some data on our results. I will have to skip because time is running. Okay, so the summary of we get for acetyl, gluco, phosphor, and exo. For all you find a structure like this. And in any of them you can apply the method and proceed with the classification. Okay? Here are some of the matrix that you present. Here it's here I use it to represent acetyl, gluco, exo, and phosphor. Okay? Here is what you get when you retrieve the matrix at different levels. In the exo, when you retrieve it at 37%, you get this picture. When you draw the matrix at 40%, you get this kind of figure, and 44 and 56%. Okay. Here the corresponding dendrograms that you obtain. And here's I have also showed you. Then we can come to the results. The number of distinct sequences that we used happened to be a little bit shorter, not 1695, 1645, because some of the sequences were fed into the data bank twice. So actually we worked with 1645. Okay. Each sequence belongs to only one network. Okay. But there are not 1645 different organisms. There were only 382 distant organisms. That means that some organisms were present in all 5 networks. Some organisms were not present in one of the networks. Some organisms were present in just one network. Okay? So that's what I said, more than one sequence can be present in the same organism. So we proceed with the congruence of classification. That we put together everything that belongs. For instance, this is we now order not by the sequences of proteins, but the organisms, the different kinds of organisms, 382. And so these are the classification. That means that all which is green here belongs to the same community. What's red here belongs to the same community. Here, we do the same thing. That does not mean that what is red here corresponds to what is red here. It could be different, because the classification and the color scheme is different from one another. But in fact, this red corresponds to this red, this green corresponds to this green. It's also important to show that some networks have a different number of communities. This led to 7 different communities, this one to 4 different communities. So a community given by fossil must entail 2 or more communities of UGP. Okay? Here it was not possible to, the structure of the protein here was not sufficient to make distinction between 2 kinds of groups that the UDP was able to make the classification. Okay? So this is the same picture that we are just spaced in order to be able to see what we get. What the void space is just because this organism is not represented in this sequence, in this network. Okay? And then the summary of what we get, the number of communities in each different, and the number of different nodes in each of the networks. And this what we call the congruence, how the classification of one given by one network fits with the classification provided by the other networks. We get, in average, a number of 84% of classification success from one network to the other network. Okay? Then we, quite recently, we also obtained some results for the chitin synthase. The chitin synthase is a molecule that we not included because it was present only in 40 organisms. And in our first approach, 40 was not enough to use a network. But we chose now this one because we wanted to compare with other phylogenetic classification based on methods that were developed by the biologists. And then Bayesian, distance, likelihood, and parsimony. There are 4 different classification schemes. And our comparison with theirs find this match, 56, 53, 58, 64. Those scores are smaller than the score that we obtain when we compare between ourselves, our results, but they are similar when those met. If you compare parsimony with Bayesian, it does not go much more than this, 65%, something like this. Okay, so that we are somewhat playing in the same kind of quality of the results, and so this is the last slide where we draw our comparisons. Now, which is a synthesis of what I said. We used a network approach to process some kind of biological data with no biological information. That's an important issue. We make no assumption that this organism is related to the other one. The only information that we used was the similarity between the proteins. We make no information at all. If blasts use some kind of information, we do not know. But blasts for us is a black box. We just took blasts and used to proceed with the similarity. And then I think we are very amazed to find that our classification scheme was able to provide some success in the good approach. Thank you very much.
Thank you, Roberto. Questions or comments?
I have a question. How do you decide which enzyme that you want to take into account to then construct the network in such a way that they have the same number of nodes, which are the proteins
we chose to work with chitin synthesis. Now we are working with 2 more input data, which are the glucose and the ATP. Okay. Then when we chose the chitin, we scan the database looking for proteins that are present, enzymes that are present in the synthesis of the chitin. Then that's where we found those 1695 different types. Okay. Then we identified which were UDP, which were acetyl, and so on. The number is the number that is in the database. The number that is not the length of the amino acids. The number is the number of sequences that have been identified by other researchers. Then we have 176 different protein sequences that are related to acetyl. Okay. So that's how it. Then we proceed. Acetyl, use acetyl, and use blast and determine that score that says how acetyl of organism A is similar to the acetyl enzyme produced by organism B. Okay, they give it a score. So it's not that we have to choose this. The network size, it depends on the data that you work with.
And those enzymes that may be in the same pathway, but they are different when the organisms are different. So for example, glucolysis, you will find different enzymes if you look the glucolysis at different organisms. If you just
I think that you are going to identify some enzymes that are present in the synthesis of glucose, and they represent the same.
So you already take only those that are present in both.
Yes, in many organisms. Yes.
You are considering already those similar.
Yes. We look when in the database it is possible to identify that sequence of amino acids is identified as a UDP protein. Okay. A UDP enzyme, okay, and then you take okay, this is a UDP enzyme, we put it in our bag. Another organism has another UDP enzyme, we put our, if it's not UDP, we do not put in that bag, we put in other bag, perhaps. Okay.
More questions or comments? I have a philosophical question. Your method is mostly based on it's a kind of distance method you have compared with several ones. So you classify organisms as in the same cluster as if they have similar protein pathways. Another philosophy is that say more evolutionary, as the maximum likelihood or the Bayesian method is stated to fit the sequence you see to a model of evolution and see which evolution is more consistent with the sequence you see at the end. In my opinion, this would reflect better, or is more oriented to the history, to the evolutionary history. Then my question is: do you have any idea, any clue how to distinguish in your method similarities which are accidental and in different evolutionary pathways give to the same pathway, or just it's as the distance method and all are not bad at all?
We did not use it at all. We have two biologists in our team, and they were the guys that proposed us to look if it was possible to devise a method for finding phylogeny without biology and knowledge. Okay, so that's really the important issue that you make very clear. We do not use no biological assumption, no at all. It's only based on the similarity of the proteins. Which one is the best? I think that phylogeny is an open question. It's not easy to define which the right. As I told you in one of the last slides, if you compare Bayesian with distance, Bayesian with likelihood, the results that they propose, and if you compare with them, is not much greater than the data that we obtained. So it's really a very difficult and unsolved question, how to really provide a fine classification. So we proposed that one. Let's see if the biologists accept it or not. Let's see. I'm excited about see what's going to happen.
Any other question? If not, thank you again.